Hi everyone, in this video we will explore reference-based single-cell annotation methods. So in the past video we have seen the marker-based approach where let's say you take your query and you have the marker genes and you query against some databases, marker databases, and based on that you try to transfer the cell type information to your current dataset. So this is the second approach where let's say you have your query data and you have a reference data by let's say someone has done some experiment and they have the single cell data and they have done the hard work and they know like okay in their data set these are the cells and they belongs to let's say this cell type 1, these cells belongs to cell type 2. So let's call that a reference data. So that means we have some reference against which we can compare our dataset to their dataset, and based on that, whatever the if some match is happening, then we will transfer that knowledge. This is very much similar as our sequence alignment, right? So you have the reference genome, and we take the reads and try to map it, and we align our sequence against the reference sequence and see how much similarity are there, right? So the same approach is this all this method works. That's what we will see. So you take a query data set and let's say some reference data set is there where already clusters, you know what levels they are, and you just basically compare these two and transfer that knowledge to your query data set, and finally you annotate it. So that's how the level transfer occurs compared to the reference. And here statistical method comes into picture because all this comparison happens via statistics, and there are several tools, several approaches are there which you can broadly categorize into three categories. Like for example, the SingleR is a very widely used R programming R package, which is basically correlation based, Spearman correlation based. It try to correlate your test data against that reference data, and based on that it transfer that knowledge from the reference to your query. The second approach is mainly projection based. This is like one tool is falls under category Azimuth. This is developed by the same team who have developed Surat. So here projection means in the PCA space of reference data you try to project your query, and based on that wherever your query goes, try to fit or project it into the projection space, you transfer that knowledge. In third category of models or tools you will find is mainly machine learning models. That means they are already been trained to classify into different this cell type, that cell type, like that. So there are various tools. That's why those are semi-supervised models are available. And for example, tools like CellTypist and scRNA-seq, and there are some other tools are available which falls into the pre-trained ML model category. And for example, CellTypist uses logistic regression and scRNA uses some other ML based approach, and based on that it will help you to classify. So in this video we will first focus on the SingleR, which is widely used in the correlation based approach. Okay, so that's what we will see. Okay, so before that, right, so now for all this approach what we need a reference data set. So now you might ask like from where we will get it, right? So there are two things, two ways you can get it. One is like for example you can use some already existing package like Celldex package, which has pre-compute pre-built data sets are there. We can use that, leverage that package to compare initial guess. Or alternately you can read some paper, and if you get some like closest match to your query to that reference paper, you can get that reference data like gene by the sample, right, the cell count matrix. If you can get, then you are good to go, and you can use the SingleR package to do that correlation transfer as well. So this is the one of the most challenge like to getting the single cell reference data. And there are many database are available as well, which you can get for that main reference data set. But Celldex package is latest, I think updated 2024, that includes many widely used data set like HPCA, human I forgot the full form name, some cell atlas, okay, and blueprint and code dice, those kind of thing. Okay, I think we can check what is that. This is the Celldex package I am just showing. If you go to in our red packages and you can go to Celldex, I have already installed it, that's why I can see it. And so here we can go like for example, yeah, HPCA is human protein cell atlas. Okay, so all the data sets are there. If you click here, so all the data from HPCA is available in this package. Similarly for mouse, for human, there are different data sets are available. So like I think 1, 2, 3, 4, 5, 5 from human and 2 from mouse. And how many samples the data is there, that is also there, and the technology, the microarray and bulk RNA based, and what cell types it will fit. Okay, so this is a very nice package, and SingleR package is a tool that also suggests to use this package. So just for quick start. Okay, as I said, alternately, if you don't want to rely on this, you yourself can create that matrix. I will talk about how to create that and how to do that, but I will start with the Celldex package. So what is this? Is our package for single cell reference dataset. Okay, and the range from bulk RNA to microarray, and the data is mainly in the format that SingleR expect. SingleR expects your reference data should be log normalized, means it should be normalized across cells. Also the value should be log transformed. Okay, I will tell you why it needs that in the next subsequent slides. But so the data what we need is like log normalized expression matrix, where the rows will be the genes and columns will be the cells. Okay, and now for each of this data set, it provides 3 level of annotation. The label.main means it's more broad annotation. So basically it covers major cell types, but the resolution is very low. But because very less number of levels, so if you use this category to categorize your annotate, it will be more faster. Then the second category, level.fine. So it's again fine grained annotation. So where you can go up to subclass and subtypes. So that's why it is little comparatively slow, but it gives little better resolution. The third is same as level.fine, but along with the subtypes, it will also give more controlled vocabulary in the form of cell ontology terms it gives. So usually to begin with, we can use level.main, and then to further, let's say in a scenario where 3-4 clusters are merged and annotated as, let's say, T cell, then you can go for level.find to further differentiate that big lump cluster. That approach also hierarchically you can follow. Okay, so yeah, that is the one thing, Celldex. Now next come to SingleR. So how does it work actually? This is a little bit I'll try to explain how it is. So now let's say this is your reference data. Okay, so you choose any of the reference data. So in reference data, the format would be gene by samples. Okay, so let's say these are the some 10,000 genes, something like that, and this is sample 1, sample 2, sample 3, sample 4. Sample means cell here, cell 1, cell 2, cell 3, cell 4, like that. And let's say this 4 belongs to cell type A. This is, let's say, B cell. This is some other cell. So we have categorized. So this way, let's say we have this gene by sample or cell matrix, and we have the cell type information. So this is what the reference data will look like. So what first SingleR does, like, see, there are so many genes would be there, right? Because this is reference data set. So why to work with this many genes? So first what it does, it will try to compare the same, how Surat you take this cluster and do the differential gene expression and find the marker genes. So first what it does, it in the reference data set, it will try to find only the top differential expression genes. So now let's say from 20,000 you got, let's say, 200 genes which are differentiating across the cell type. So now in the downstream analysis, only those 200 genes will be used. So that's the reason why the data what SingleR expects is in the log or normalized data. Okay, because it is comparing the each cell type to first identify the differential expression gene. So that's why that is the main thing, like the format expectation from SingleR is that like your matrix should be in the log normalized format. Okay, so now let's say we have filtered it. So now we got selected differential expression genes. Let's say hypothetically 2 genes here, and the same format, sample 1 to sample n, and we have the category like cell type 1, cell type 2, cell type 3, like that. So now this is what our reference data set first we got. Okay, so now the second is your query data set. So in the query, let's say we have the same number of genes and you have cell 1, cell 2, cell 3, but you don't know what they are, right? So we do not have that annotation. So our query matrix will be, let's say, genes by sample or genes by cells. Okay, so now what you do, you take first cell here. So now you take this cells, let's say we want to annotate this particular cell number 1. So what SingleR does, like it takes this cell and it tries to find the correlation between this first column here with the first column. It's cell 1 with that sample 1, cell 1 to that sample 2. So like that, for every sample in the reference dataset, it tries to find the correlation. So what you will get now, for this particular cell 1, you get how many correlation? That means SN correlation, right? For every sample, you will get one correlation, S1 to SN. So now what you do in the next step, what it does, it sorts them and select the 80% cutoff it chooses. Just I will give an example. So in this case, let's say cell type 1, if you chose cell type 1, how many samples are there? We have 4 samples, S1, S2, S3, S4. So now we are taking this cell 1 and now we are doing the correlation, Spearman correlation we are doing with the S1, S2, S3, S4. So how many scores we'll get? 4 scores, right? So let's say with respect to the sample 1, the cell type, the score is 0.82. For sample 2, the correlation is 0.75, 0.773, sorry, 0.82 and 0.88. So now what you do, you sort it and you took the 80% percentile cutoff you choose, and whatever the cutoff value you will get, so that score, that is the cell type A score. So now you assign a score, that score you assign to here for that cell 1 with the type 1, okay, type A, because we have 4 sample we took, right? So in the second phase you take again cell 1, now compare with S5, S6, S7, S8 and do the correlation, sort them, select the 80% cutoff, and that score you assign to the second 0.5. So now by repeating that process for that cell 1, now you got score from score against type A, type B, type C, type D, like that. So for every cell type in the reference data set, you get a score. And score is simply by the aggregation of the 80 percentile of the correlation coefficient. Okay. So now we will get, like, finally we get this matrix. Like for every cell type we get the score against all the cell types. So kind of matrix. So let's say in the reference we have 10 cell type, and you get number of cells into 10 metric dimension you get. So now this is the most important part. So now what it does, like cell number 1, by default it directly cannot assign, do not assign type A, because being highest, what it does, like it does something called fine tuning. So what it does, like it for every cell it will now take a top two cell type, let's say type A, type B. So now it will do an iterative process. What it will do, it will now iterate this process, it will come back again. So here now the reference dataset will now contain type A and type B. So basically it will subset this reference dataset, and it will again find the differential gene expression between those two cell type again, and it will repeat the process. So now just imagine in the very beginning, we when we started with the analysis for this cell type, we took the entire matrix, right? We took the entire matrix and we are comparing, and we did the process and got the two cell type. Now you are reiterating it. So instead of that, now you are just taking this many data set, and because of that your differential expression gene will change, because earlier in the first iteration your differential gene expression of cell type A was against cell type A versus everything together, right? So now because you are now the differential expression will change between A and B. I think you got the point. So that's why it iteratively does the process, ultimately fine tuning it does that only highest score it gets. Okay, that is the the logic SingleR works that way. Okay, so just to again reiterating, like it works on it takes the queries your gene by cell and compare it against gene by sample, okay, labeled sample, and it uses Spearman correlation because it doesn't use, it says rank based, right? So that's why even if your values, it's mostly non-parametric, right? It is rather than, I mean, it is very resistant to noise and batch effect, because you are not considering mean, median, mode, those kind of data shape. Not, you are not considering any statistics here. Basically you are ranking the expression, and based on that you are comparing. So if a gene is highly expressed in your query and the gene is highly expressed in even reference, that means they are rank wise they will be on the top and they will be highly correlated. But if a gene is positively regulated in its query but very less expressed in the reference, so those are negatively correlated, right? So it will, the rank is very high. So that way, based on the rank correlation, it compares query versus the reference, you will get a correlation value. Okay, so that's the another point, like instead of looking at all those 20,000 genes, we consider first initially the variable genes, and the differential expression genes also we consider, or it considers. Okay, so now this already I have explained, like for each test how it calculates, then again it does, so I will not repeat here again. But now this is the most part, the fine tuning. Oh, I already explained, like first it will starts with all possible level, right? Then it does the pruning, it reruns the correlation with only taking the top remaining levels, let's say top 2. Then it will again repeat until finally one level will remain, and then it will give you something called delta score. Okay, that's a very important part. So what delta score is nothing but the what it like, so you get a score between, for every cell type, right? So the score difference between the top 2 is called the delta score. Okay, that gives the confidence. So higher is the delta, means the difference between the score against the top versus second top is very high. So that means you are more confident behind the call. Okay, so that's what we should look for, the delta score in the SingleR that gives the confidence behind this annotation levels. Okay, so without that, with that introduction, let's go back here. So here is the SingleR book. Very nicely they have documented, so you can just read through. Okay, so we'll start with the practical. Okay, so let me start with the practical just to show you. So we need to install SingleR, Celldex. These are the all dependency we need to install. Let's load it. So I'm just loading all the required library. So first let's explore the Celldex. Okay, just we need to understand about the reference. So if I go to Celldex package here, from packages also you will be able to find it, and there is two functions are there. One is called survey references. If you call that, it will give you the what they are. So as I said, there are 7 data sets are there, and what those data set mean by. Okay, so this is one way to know what the PubMed paper and the URL, those kind of thing, extra information you can get it. Okay, alternately you can just see the name. If you call this function list references, it will give the name like HPC. But why would suggest what I would suggest, like, you can go to the package folder and you can choose any particular data set and read like what they are and how does it, what it contains, which paper they took from, like that. Okay, so now in this example, the PBMC dataset, we will what we will do, we'll take this human cell atlas data, and against that we will compare it. Okay, so you simply call that function, and yeah, and you can also choose further parameter, but I will choose the default. Now this is the reference dataset. Okay, so let me just, so now if I just type ref, right, so this is the reference data set. The data is in the form of summarized experiment class. Okay, and the dimension is this many genes and this many samples are there. Okay, and as I said, the data is in log counts. Okay, which is what SingleR expects, log normalized data. Okay, and the gene names are on the rows, and the column names or the sample names are the GSM IDs. Okay, and the metadata is like 3 metadata. Label.main means broad category, fine means fine tune, and the corresponding ontology, 3 category it is there. Okay, so you can just look for the column data. So if I do that, it will this is what the column data means. 713 samples are there, right? And this is the label.main, the DC category, and this is the fine level DC dot monocyte derived, and the ontology. This is what you can take that ontology and query against the ontology in the you can query this ontology to get the further information. Okay, so this is how the metadata about the cell or the samples. If you want to see the further, right, so what we can do, we want to see how many categories are there. Okay, so I'll just group by this and I'll just show you what it means. Like, so in this data you can see that we have so many uh, cell types are there. For example, BM prognostic we have 1. Okay, astrocyte we have 2 samples, and myeloid 2 samples. But if I see for the macrophages, 90 samples that are there. 90 in that matrix, 90 samples are there, which just show you. So in this matrix, S1 to S90 is, let's say, your macrophages. It samples belong to DC. So like that we have see, these are the rare, you can say that these are the rare samples. So that's what, you may not get it, or if you get it, then you have to rely on only one single sample. You have to, the confidence might be low. Okay, so that's the dish. That's why any data set you choose, first you see like what kind of cells are there and how many samples are supporting that particular cell type. That's a very important parameter that we should look for. And just to see the log counts, it is in the log scale. The levels will be not there because that's a single cell format. Okay, so now so we got this reference data set. Okay, so that is there in this variable called ref. So now what you do, you just run your PBMC pipeline. Suppose Surat pipeline you run, and just load it, find these all are normal C pipeline. So now you just need to convert this PBMC. This is PBMC is in the format of the Surat object. So we need to convert that into single cell experiment object. So that's the requirement for SingleR. Okay, so let it run. Okay, so now if I just check, what is the class of PBMC, this is a surrogate object. Okay, so we need to convert that into single cell object. So we can call this function age.SingleCellExperiment, and this is the part of surrogate package. So if you convert that, it will auto converts, it will give some warning. But if I just say class of SCE, it says that it's a single cell experiment object. Okay, so now in the next step, what we simply call this function, and you give what is the query. So in this case, our query is this SCE data object, which is the PBMC query, and the reference is the human cell atlas. Okay, and here what currently I'm choosing is the main level dot means the broad category. I want to do that, and this is any additional parameter you can pass the surrogate cluster information. Just to see what's the cluster means, it will say that this barcode belongs to cluster 0, this one belongs to cluster 1. So that cluster information it will give, so that it will try to map that into the whatever the output we get for every cell, it will map it. Okay, so let's run this. So it runs very fast because it's just correlation, so it runs very fast. It will maybe for larger data set it may be very slow. Okay, so first let's see the labels what you got. This is the prediction of the main object, which is now our input data is now labeled. So we see that label from T cell, B cell, broad category, and I think, and I could see 1, 2, 3, 4 clusters marked with the T cell. Okay, so that's what I was saying, like broad category will give you in big cluster. So we'll see like how does it look like. And so this is what you see, that one cluster, 4 clusters belong to T cell, and 1 cluster platelet, 1 cluster NK cell, and again 4 clusters belong to monocyte, and 1 cluster labeled as B cell. Okay, so now there are some interesting plots that further you can diagnostic plots that we should do. That's called this one, heat map of the score. Okay, so let me explain. So as I said, what we'll get is we get a score of every cell against every cell type, right? So this matrix is what you are doing, and you are doing a heat map. Okay, so when I do that, so it is looking like this. Okay, so let me directly show from here because I have labeled marked it. Okay, this plot will look like this. Okay, so the each column is one cell. Okay, so you have clustered your cells, and these are your reference levels. Okay, so, and this is on the top, it will try to also give you the final level that it has been assigned. And so for example, let's say this cluster is this color, right? T cell clusters. Okay, so now this particularly, these all bunch, right? Though this entire block, these all cells have been marked, labeled as T cell, and you can see that their expression is very high. High is the yellow one, towards the T cell. Okay, so that's how you could see, particularly during wherever T cell is there, in that you should see some yellow block. So that is the indication that it's very confident, and you should not see any yellow anywhere else. So that's contrasting, you should be able to see. The second, for example, if I say this cluster, right, let's go with this green one. So green is what? NK cell, right? The green label here. So that's why in the NK cell only you see this yellow band. Okay, so that's what the second thing. So now this cyan one, that's a monocyte, and you see a yellow patch over here. So that means these 3 are monocyte, pre-B cell, and HSC, some other name is there, but still the monocyte, this section got you got a yellow patch. And for example, this is the B cell, right? The last one, orange one, the B cell, and you can see a yellow mark here, and nowhere else yellow is there. So that gives you more confidence that okay, whatever the assignment we got is a good one. Okay, so that's what you can see in the vertical line. You see that ideally you want to see one level, or that means one particular row with a significantly higher color, means yellow, then all other the rows, that kind of thing you should see. So this is a very interesting plot, an important plot, I would say, that will help you to see how this distribution will look like. Okay, and this is, if you do the normalization false, means it will give you another kind of information, like but you should do the normalization, you should not remove this one, but if you want to try that. So now then something is called delta distribution. This is a very important plot, because as I said, we get 2 scores, right? The first and the second best, and the difference is the delta. So high is the delta, means you are more confident that assignment is high. Okay, so that's what we will see when I do that, right? So there are 2 approach we can do. One is what kind of delta you want. Okay, there is something 2 delta. One is delta next, and another called delta median. So delta next means like this, the top one minus the next one. Okay, so that is the delta next. Okay, the first 2, but The delta median is the difference between the score of the assigned level and the median of the all scores for the each cell. Okay, one versus all the other cell types. That kind of the median you do. Okay. So for example, if you just look that, so we got a B cell, right? And as I said, for B cell, we got how many distribution? B cell only one cluster belong to that B cell. Okay. And that confidence is 0.1, means low delta. So that means we are confident. Now, if you come to monocytes, right? Monocyte, we had 4 clusters got projected. Okay, just let me do one thing. First I will do the heat map. So not heat map, the UMap plot. So first let's do that, then I can explain. So for that, let's say we are going with this level what we got. So you can simply assign the levels, and this rename identity you do, and you assign your object with that, and just I'm storing the metadata, I'm visualizing. Okay. So now you see that this particularly block entirely got the T cell, and monocyte is this green one. Okay, because I have done the broad category. Okay, so now this, you see 4 clusters are over here which are monocyte, and so that's why you see 4 dots, and of which one is very less, delta is very low, delta is very oh, okay, my bad. I think so here, my bad, the B cell delta was very low. I said delta is very low means confident, my bad. So what I mean to here, so here delta is low means here B cell assignment is a little less confident. Okay, because delta is low. Okay, there is the B cell versus the next one is very close. Okay, so we should not again, platelet is also less confident, and here for monocyte, for one of the clusters we got high confidence like that, and here we have low confidence here. Okay, but B median, you get some higher value here, means like the median one in the 0.35 range. Yeah, here also in 0.3. So I don't know what scales to choose. The recommendation value that we can I have to see. Okay, what value they are recommending. Let's check diagnostic plot. So I don't know whether they have given or not. Yeah, they have given these plots only. Let's say measure the delta scores across cell. This one I wanted to check. Sorry, my network is slow, I think. Yeah, this is the delta plot what we got from this example. So we can use this plot to check the outlier detection. It is saying not that. See, our assumption is that most of the levels in the reference are not relevant to any given cell. Thus, the median across all the level can be used as a measure for the baseline correlation. So, okay. So again, that's what, even if what they're saying, like, the delta is really low, but since it's not a direct correlation, right? So you will be getting very less score over here. So that's what the median value you can choose rather than this top, this one. So maybe delta dot median will give the good indication. Okay. If the cell's true level does not exist in the reference. Yeah, so low delta indicate that the assignment is uncertain. This is the notes I got from their website. Low delta indicate that the assignment is uncertain, which is especially relevant if the cell's true level does not exist in the reference. Okay, so yeah, so why this plot distributes delta distribution, this function, right? It did not run, so let me try that. So we saw this one heat map. Now this is the delta distribution. So this is the delta next plot, and here you can pass the delta median. Okay, and let's read it from there. If I go to SingleR, right? Okay, so I'm just looking for this plot distribution, plot delta distribution. Okay, there are some other parameters are also there. You can try it out. Whether we need to do pruning or not, display pruning or not, so those kind of things. Okay, but in short, right, what I mean to say, like, this plot will help us to where the weak spot weak annotation, like, for example, if a specific cell type NK cell has a very low median delta compared to others. Okay, so those assignments are shaky. Okay, so that's what this assignment is very less confidence. But I think if we go with this fine tuning, right, so currently this is the B cell, right, only broad category. But if you go with probably the fine grain, we might get confidence score should be high. Okay, so now this is the most important part, where how will you, let's say this Celldex dataset is not available. So then if you are using any reference data custom, so you just make a matrix gene by cell, and the data you just do a log transformation. That is the requirement, because with that log transform data is used for the calculating the differential gene expression information. Okay, and for test dataset, no need of log transformation, because we are using correlation, and that's the information it shows, like because correlation, so we do not need an actual expression value. Okay, and yeah, choice of the reference means like very important. You should select a reference that contains a superset of all the cell types you expect to find in your test dataset. Okay, that's why your reference should be broad. Okay, so then only you will be able to do that. That's the one recommendation. And the accuracy of your results will depend on the quality of the original author's level, right? So some references are naturally better than the other due to superior sample preparation. So that's the thing, like cell reference based will depend on how accurate your reference data set is. Okay, and third thing is like technological compatibility. That means it is ideal to use a reference generated from a similar technology and protocol. Someone like microarray is microarray, but it's very rare. So you can go with this approach. But yeah, and yeah, so log expression value I've talked. Okay, so with that, I think SingleR is a very nice package. I would recommend you to go through this thing, their books, and they have some other information as well. For the details, it would be very difficult to cover everything, but this gives you an idea. Method is very simple, one line command, but concept is like the reference what you're using must be log transformed, and then your query is anything is fine. You just compare and do the correlation based analysis and do some plotting, and that will give you information. And at the same time, you can take that and now compare against marker as well, and try to see and make a consensus approach as well. So you have to use multiple method to come up with the final answer. Okay, so that's all on about this SingleR one approach. So another one is Azimuth, which is developed by Surat team. So let me just show you. Azimuth, I think this is the one. Unfortunately, this app is not working on the UI, and yeah, it is very it's someone not loading, and I'm struggling with the installation in my system. So I will see if I can make a video about it. But if you get a chance, definitely you can try to explore this one. And the GitHub page is this one, Azimuth. You can, sorry for that. And see, it's it's hanged. I'm not able to do that. Okay, Azimuth GitHub. If you just type that one, this is what it's a shiny app, and you can try to install and do that. And they also provide in the Docker format. I also tried to do the Docker, but somehow in my system it is not working as of now. I'll have to spend some more time to make it work, and so that I can explain. Earlier, it was working, and when I was exploring, it was working very fine. But right now it is not working. Okay, so with that, we will move to I will next move to the CellTypist, the ML based. So we saw one SingleR. Azimuth, I will try to do that, but CellTypist is one logistic regression based, and I will cover that. Will give you like how ML based approach it will work. And this is mainly, the CellTypist is mainly Python based, so I will switch to Python, but that will give you some idea about how ML based method works. I hope this video was useful, and thanks for watching, and I'll see you in the next video. Please do provide feedback in the comment section, like how I can improve, and if you want to know more about any other topic, it will help me and it will boost the channel. Thank you so much. Thanks for watching. Bye bye.